We continue from where we stopped in the last lecture. We're on to the next topic, B2, the nucleus. The presence of a nucleus is the principal feature that distinguishes eukaryotic from prokaryotic cells. By housing the cell's genome, the nucleus serves both as a repository of genetic information and as the cell's control center. DNA replication, transcription, and RNA processing all take place within the nucleus, with only the final stage of gene expression, translation, localized to the cytoplasm. By separating the genome from the cytoplasm, the nuclear envelope allows gene expression to be regulated by mechanisms that are unique to eukaryotes, whereas prokaryotic mRNAs are translated while their transcription is still in process. Eukaryotic mRNAs undergo post-transcriptional processing, example splicing, before being transported from the nucleus to the cytoplasm. The presence of a nucleus thus allows gene expression to be regulated by post-transcriptional mechanisms such as alternative splicing. By limiting the access of proteins to the genetic material, the nuclear envelope also provides novel opportunities for the control of gene expression at the level of transcription. For example, the expression of some eukaryotic genes is controlled by the regulated transport of transcription factors from the cytoplasm into the nucleus, a form of transcriptional regulation unavailable to prokaryotes. The separation of the genome from the site of mRNA translation thus plays a central role in eukaryotic gene expression. The nuclear envelope and traffic between the nucleus and cytoplasm. The nuclear envelope separates the contents of the nucleus from the cytoplasm and provides the structural framework of the nucleus. The nuclear membranes acting as barriers that prevent the free passage of molecules between the nucleus and the cytoplasm maintain the nucleus as a distinct biochemical compartment. The sole channels through the nuclear envelope are provided by the nuclear pore complexes, which allow the regulated exchange of molecules between the nucleus and cytoplasm. The selective traffic of proteins and RNAs through the nuclear pore complexes not only establishes the internal composition of the nucleus, but also plays a critical role in regulating eukaryotic gene expression. Structure of the nuclear envelope. The nuclear envelope has a complex structure consisting of two nuclear membranes, an underlying nuclear lamina, and nuclear pore complexes. The nucleus is surrounded by a system of two concentric membranes called the inner and outer nuclear membranes. The outer nucleus membrane is continuous with the endoplasmic reticulum, so the space between the inner and outer nuclear membranes is directly connected with the lumen of the endoplasmic reticulum. In addition, the outer nuclear membrane is functionally similar to the membranes of the endoplasmic reticulum and has ribosomes bound to its cytoplasmic surface. In contrast, the inner nuclear membrane carries along unique proteins that are specific to the nucleus. The critical function of the nuclear membranes is to act as a barrier that separates the content of the nucleus from the cytoplasm. Like other cell membranes, the nuclear cell membranes are phospholipid bilayers, which are permeable only to small nonpolar molecules. Other molecules are unable to diffuse through the phospholipid layer. The inner and outer nuclear membranes are joined at nuclear pore complexes, the sole channels through which small polar molecules and macromolecules are able to travel through the nuclear envelope. Underlying the inner nuclear membrane is the nuclear lamina, a fibrous meshwork that provides structural support to the nucleus. The nuclear lamina is composed of one or more related proteins called laminins. In addition to providing structural support to the nucleus, the nuclear lamina is thought to serve as a site of chromatin attachment. The nuclear pore complex. The nuclear pore complexes are the only channels through which small polar molecules, ions, and macromolecules, proteins, and mRNAs are able to travel between the nucleus and the cytoplasm. The nuclear pore complex is an extremely large structure with a diameter of about 120 nanometers and an estimated molar molecular mass of approximately 125 million daltons, about 30 times the size of a ribosome. In vertebrates, the nuclear pore complex is composed of 50 to 100 different proteins. This diagram is indicative of that. By controlling the traffic of molecules between the nucleus and cytoplasm, the nuclear pore complex plays a fundamental role in the physiology of all eukaryotic cells. RNAs that are synthesized in the nucleus must be efficiently exported to the cytoplasm, where they function in protein synthesis. Conversely, proteins required for nuclear functions, example transcription factors, must be transported into the nucleus from their sites of synthesis in the cytoplasm. In addition, many proteins shuttle continuously between the nucleus and the cytoplasm. The regulated traffic of protein and RNAs through the nuclear pore complex thus determines the composition of the nucleus and plays a key role in the gene expression. Depending on the size, molecules can travel through the nuclear pore complex by one of two different mechanisms. Small molecules and some proteins with molecule mass less than approximately 50 kD pass freely across the nuclear envelope in either direction, cytoplasm to nucleus or nucleus to cytoplasm. These molecules diffuse passively through open mucoaqueous channels, estimated to have diameters of approximately 9 nanometers, in the nuclear pore complex. Most proteins and mRNAs, however, are unable to pass through these open channels. Instead, these macromolecules pass through the nuclear pore complex by an active process in which appropriate proteins and RNAs are recognized and selectively transported in only one direction, nucleus to cytoplasm, or to cytoplasm to nucleus. The traffic of these molecules occurs through regulated channels in the nuclear pore complex that, in response to appropriate signals, can operate to a diameter of more than 25 nanometers, a size sufficient to accommodate large ribonuclear protein complexes, such as ribosomal subunits. It is through these regulated channels that the nuclear proteins are selectively imported from the cytoplasm to the nucleus, while RNAs are exported from the nucleus to the cytoplasm. Visualization of nuclear pore complexes by electron microscopy reveals a structure with eightfold symmetry organized around a large central channel, which is a route through which proteins and RNAs cross the nuclear envelope. Detailed structural studies, including computer-based image analysis, have led to the development of three-dimensional models of the nuclear pore complex. These studies indicate that the nuclear pore complex consists of an assembly of 8 spokes arranged around a central channel. The spokes are connected to rings at the nuclear and cytoplasmic surfaces, and the spoke ring assembly is anchored within the nuclear envelope at sites of fusion between the inner and outer nuclear membranes. Protein filaments extend from both the cytoplasm and nuclear rings, forming a distinct basket-like structure on the nuclear side. The central channel is approximately 40 nanometers in diameter, which is wide enough to accommodate the largest particles available to cross the nuclear envelope. It contains a structure called the central transporter, through which the active transporter of macromolecules is thought to occur. Internal organization of the nucleus. The nucleus is more than a container in which chromatin RNAs and nuclear proteins move freely in aqueous solution. Instead, the nucleus appears to have an internal structure that organizes the genetic material and localizes some nuclear functions to discrete sites. The most obvious aspect of the internal organization of the nucleus is the nucleolus, which, as discussed in the following section, is a site at which the RNA genes are transcribed and ribosomal subunits are assembled. Additional elements of internal nuclear structure are suggested by the organization of chromosomes and by the potential localization of functions such as DNA replication and pre-mRNA processing to distinct nuclear domains. The nucleolus. The most prominent substructure within the nucleus is the nucleolus, which is the site of rRNA transcription and processing and of ribosome assembly. As discussed in the preceding chapter, cells require large numbers of ribosomes to meet their needs for protein synthesis. Actively growing mammalian Cells, for example, contain 5 million to 10 million ribosomes that must be synthesized each time the cell divides. The nucleolus is thus, or is, a ribosome production factory designed to fulfill the need for large-scale production of RNAs and assembly of the ribosomal subunits. The nucleus during mitosis. A unique feature of the nucleus is that it disassembles and reforms each time most cells divide. At the beginning of mitosis, the chromosomes condense, the nucleolus disappears, and the nuclear envelope breaks down, resulting in the release of most of the contents of the nucleolus into the cytoplasm. At the end of mitosis, the process is reversed. The chromosomes decondense and nuclear envelope reform around the separated sets of daughter chromosomes. The process is controlled largely by reversible phosphorylation and dephosphorylation of nucleoproteins resulting from the action of the CDC2 protein kinase, which is a critical regulator of mitosis in all eukaryotic cells. The next topic will be B3, specifically on mitochondria. Mitochondria play a critical role in the generation of metabolic energy in eukaryotic cells. They are responsible for most of the energy derived from the breakdown of carbohydrates and fatty acids, which is converted to ATP by the process of oxidative phosphorylation. Most mitochondrial proteins are translated on free systolic ribosomes and imported into the organelle by specific targeting signals. In addition, mitochondria are unique among the cytoplasmic organelles in that they contain their own DNA, which encodes tRNAs, rRNAs, and some mitochondrial proteins. The assembly of mitochondria thus involves proteins encoded by their own genomes and translated within the organelles as well as proteins encoded by the nucleic genome and imported from the cytosol. Organization and function of mitochondria. Mitochondria is surrounded by a double membrane system consisting of inner and outer mitochondrial membranes separated by an intermembrane space. The inner membranes form numerous folds, cristae, which extend into the interior matrix of the matrix of the organelle. Each of these components plays distinct functional roles, with the matrix and inner membrane representing the major working compartments of mitochondria. The matrix contains the mitochondrial genetic system as well as the enzymes responsible for the central reactions of oxidative metabolism. The oxidative breakdown of glucose and fatty acids is the principal source of metabolic energy in animal cells. The initial stages of glucose metabolism glycolysis occur in the cytosol, where glucose is converted to pyruvate. Pyruvate is then transported into mitochondria, where its complete oxidation CO2 yields the bulk of usable energy, ATP, obtained from glucose metabolism. This involves the initial oxidation of pyruvate to acetyl-CoA, which is broken down to CO2 via the citric acid cycle. The oxidation of fatty acids also yields the acetyl-CoA, which is similarly metabolized by the citric acid cycle in the mitochondria. The enzyme of the citric acid cycle located in the matrix of mitochondria thus are central players in the oxidative breakdown of both carbohydrates and fatty acids. The oxidation of acetyl-CoA to CO2 is coupled to the reduction of NAD plus and FAD to NADH and FADH2 respectively. Most of the energy derived from oxidative metabolism is then produced by the process of oxidative phosphorylation, which takes place in the inner mitochondrial membrane. The high energy electrons from NADH and FADH2 are transferred through a series of carriers in the membrane to molecular oxygen. The energy derived from these electron transfer reactions is converted to potential energy stored in a proton gradient across the membrane, which is then used to drive ATP synthesis. The inner mitochondrial membrane thus represents the principal site of ATP generation, and this critical role is reflected in its structure. First, its surface area is substantially increased by its folding into cristae. In addition, the Inner mitochondrial membrane contains an unusually high percentage, greater than 70% of proteins, which are involved in oxidative phosphorylation, as well as in the transport of metabolites. For example, pyruvate and fatty acids between the cytosol mitochondria. Otherwise, the inner membrane is impermeable to most ions and small molecules, a property critical to maintaining the proton gradient that drives oxidative phosphorylation. In contrast to the inner membrane, the outer mitochondrial membrane is freely permeable to small molecules. This is because it contains proteins called porins, which form channels that allow the free diffusion of molecules smaller than about 6,000 daltons. The composition of the intermembrane space is therefore similar to the cytosol, which, with respect to ions and small molecules, consequently, the inner mitochondrial membrane is the functional barrier to the passage of small molecules between the cytosol and the matrix and maintains the proton gradient that drives the oxidative phosphorylation. The genetic system of mitochondria. Mitochondria contain their own genetic system, which is separate and distinct from the nuclear genome of each cell. Mitochondria are thought to have evolved from bacteria that developed a symbiotic relationship in which they live within larger cells, endosymbiosis. This hypothesis has recently been substantiated by the results of DNA sequence analysis, which revealed striking similarities between the genomes of mitochondria and of the bacterium Rickettsia brevicii. Rickettsia are intracellular parasites which, like mitochondria, are only able to reproduce within eukaryotic cells. Consistent with their similar symbiotic lifestyles, the genomic DNA sequence of Rickettsia and mitochondria suggests that they share a common ancestor from which the genetic system of present-day mitochondria evolved. Mitochondrial genomes are usually circular DNA molecules like those of bacteria, which are present in multiple copies per organelle. They vary considerably in size between different species. The genomes of human and most of the animal mitochondria are only about 16 kb, but substantially larger mitochondrial genomes are found in yeasts, approximately 80 kb, and plants more than 200 kb. However, these larger mitochondrial genomes are composed predominantly of non-coding sequences and do not appear to contain significantly more genetic information. For example, the largest sequenced mitochondrial genome is that of the plant Arabidopsis thaliana. Although Arabidopsis mitochondrial DNA is approximately 367 kb, it encodes only 32 proteins, just more than twice the number encoded by human mitochondrial DNA. Mitochondrial genome encodes all of the ribosomal RNAs and most of the transfer RNAs needed for translation of these protein coding sequences within mitochondria. Other mitochondrial proteins are encoded by the nuclear genes, which are thought to have been transferred to the nucleus from the ancestral mitochondrial genome. The human mitochondrial genome encodes 13 proteins involved in electron transport and oxidative phosphorylation, as seen in the figure. In addition, human mitochondrial DNA encodes 16S and 12S RNAs, rRNAs, and 22 tRNAs, which are required for translation of the protein encoded by the organelle genome. The two rRNAs are the only DNA components of animal and yeast mitochondrial ribosomes, in contrast to the three rRNAs of bacterial ribosomes, 23S, 16S, and 5S. The plant mitochondrial DNA, however, also encode a third RNA of 5S. The mitochondria of plants and protozoans also differ in importing and utilizing tRNAs encoded by the nuclei as well as the mitochondrial genome, whereas in animal mitochondria, all the tRNAs are encoded by the organelle. The small number of tRNAs encoded by the mitochondrial genome highlights an important feature of the mitochondrial genetic system, the use of a slightly different genetic code, which is distinct from the universal genetic code used by both prokaryotic and eukaryotic cells. There are 64 possible triple codons, of which 61 encode the 20 different amino acids incorporated into proteins. Many tRNAs in both prokaryotic and eukaryotic cells are able to recognize more than a single codon in mRNA because of which allows some mispairing between the tRNA, anticodon, and third position of certain complementary codons. However, at least 30 different tRNAs are required to translate the universal code according to the Wobble rules. Yet human mitochondrial DNA encodes only 22 tRNA species, and these are the only tRNAs used for translation of mitochondrial mRNA. This is accomplished by the extreme form of Wobble, in which U in the anticodon of the tRNA can pair with any of the 4 bases in the third codon position of mRNA, allowing 4 codons to be recognized by a single tRNA. In addition, some codons specify different amino acids in mitochondria than in the universal code. Like the DNA of nucleic genomes, mitochondrial DNA can be altered by mutations, which are frequently deleterious to the organelle. Since almost all the mitochondria of fertilized eggs are contributed by the oocyte rather than by the sperm, germline mutation in mitochondrial DNA are transmitted to the next generation by the mother. Such mutations have been associated with a number of diseases. For example, Leber's hereditary optic neuropathy, a disease that leads to blindness, can be caused by mutations in the mitochondrial genes that encode components of the electron transport chain. I repeat that. Leber's hereditary optic neuropathy, a disease that leads to blindness, can be caused by mutations in mitochondrial genes that encode components of the electron transport chain. In addition, the progressive accumulation of mutations in mitochondrial DNA during the lifetime of individuals has been suggested to contribute to the process of aging. Mutation in one of the mitochondrial transfer RNA genes, characterized by a decrease in synthesis of mitochondrial protein required for electron transport chain and ATP synthesis, causes a disorder of muscles termed as myoclonic epilepsy and ragged red fiber disease, MERRF. So we're now on to B4, the endoplasmic reticulum, Golgi apparatus, and lysosomes. In addition to the presence of nucleus, eukaryotic cells are distinguished from prokaryotic cells by the presence of membrane-enclosed organelles within their cytoplasm. These organelles provide discrete compartments in which specific cellular activities take place. The resulting subdivision of the cytoplasm allows eukaryotic cells to function efficiently in spite of their large size, about 1,000 times the volume of bacteria. Because of the complex internal organization of the eukaryotic cells, the sorting and targeting of proteins to the appropriate destinations are considerable tasks. The first step of protein sorting takes place while translation is still in progress. Many proteins destined for the endoplasmic reticulum, the Golgi apparatus, lysosomes, the plasma membrane, and secretion from the cell are synthesized on ribosomes that are bound to the membrane of the endoplasmic reticulum. As translation proceeds, the polypeptide chains are transported into vesicles to the Golgi apparatus, from where they are further processed and sorted to the transport through lysosomes, the plasma membrane, or secretion from the cell. The endoplasmic reticulum, Golgi apparatus, and lysosomes are thus distinguished from other cytoplasmic organelles by their common involvement in protein processing and connection by vesicular transport. The endoplasmic reticulum. The endoplasmic reticulum, ER, is a network of membranes enclosed tubules and sac cisternae that extends from the nuclear membrane toward the cytoplasm, figure 9.1. The entire endoplasmic reticulum is enclosed by a continuous membrane and is the largest organelle of most eukaryotic cells. Its membrane may account for about half of all cell membranes, and the space enclosed by the ER, the lumen or cisternal space, may represent about 10% of the total cell volume. As shown below, there are 2 distinct types of ER that perform different functions within the cells. The rough ER, which is covered by ribosomes on its outer surface, functions in protein processing. The smooth ER is not associated with ribosomes and is involved in lipid rather than protein metabolism. The endoplasmic reticulum and protein secretion. The role of the endoplasmic reticulum in protein processing and sorting was first demonstrated by George Fallade and his team in the 1960s. These investigations study the fate of newly synthesized proteins in specialized cells, the pancreas, pancreatic acinar cells that secrete digestive enzymes into the smaller intestine. Because most proteins synthesized by these cells are secreted, Palade and co-workers were able to study the pathway taken by secreted proteins simply by labeling newly synthesized proteins, radioactive amino acids. The location of the radiolab proteins within the cell was then determined by an autoradiography revealing the cellular sites involved in the events leading to protein secretion. And if the cells were then incubated for a short time in media containing non-radioactive amino acids, a process known as a chase, the radiolabeled proteins were detected in their Golgi apparatus following longer periods. The radiolabeled proteins traveled from the Golgi apparatus to the cell surface and secretory vesicles, which then fused with the plasma membrane to release the contents inside the cell. These experiments defined a pathway by secreting proteins, the secretory pathway, rough ER, Golgi, secretory vesicles, cell exterior. Further studies revealed results and demonstrated that this pathway is not restricted to proteins destined for secretion from the cell wall. Plasma membrane and lysosomal proteins also travel from the rough ER to the Golgi and then to the final destinations. Still other proteins travel through the initial steps of the secretory pathway but are then retained and function within the ER or the Golgi apparatus. The entrance of proteins into the ER thus represents a major branch point for the traffic of proteins within eukaryotic cells. Proteins destined for secretion or incorporation into the ER, Golgi apparatus, lysosomes, and plasma membranes are initially targeted to the ER. In mammalian cells, most proteins are transferred into the ER while they are being translated on membrane-bound ribosomes. In contrast, proteins destined to remain in the cytosol are to be incorporated into the nucleus, mitochondria, chloroplasts, or peroxisomes, are synthesized on free ribosomes and released in the cytosol when the translation is complete. Targeting proteins to the endoplasmic reticulum. Proteins can be translocated into the ER either during their synthesis on membrane-bound ribosomes, co-translation translocation, or after their translation has been completed on free ribosomes in the cytosol, post-translational translocation. In mammalian cells, most proteins enter the ER co-translationally, whereas both co-translational and post-translational pathways are used in yeast. The first step in the co-translational pathway is the association of ribosomes with the ER. Ribosomes are targeted for binding to the ER membrane by the amino acid sequence of the polypeptide chain being synthesized rather than by intrinsic properties of the ribosome itself. Free and membrane-bound ribosomes are functionally indistinguishable, and all protein synthesis initiates on ribosomes that are free in the cytosol. Ribosomes engaged in the synthesis of proteins that are destined for secretion are then targeted to the endoplasmic reticulum by a signal sequence at the amino terminus of the growing polypeptide chain. These signal sequences are short stretches of hydrophobic amino acids that are cleaved from the polypeptide chain during its transfer into the ER lumen, endoplasmic reticulate lumen. David Sabatini and Günther Blobel first proposed in 1971 that the signal for ribosome attachment to the ER was an amino acid sequence near the amino acid terminus of the growing polypeptide chain. This hypothesis was supported by the results of in vitro translation of mRNAs coding secreted proteins such as immunoglobulins. If an mRNA encoding a secreted protein was translated on free ribosomes in vitro, it was found that the protein produced was slightly larger than the normal secreted protein. If microsomes were added to the system, however, the in vitro translated protein was incorporated into the microsomes and cleaved to the correct size. These experiments led to a more detailed formulation of the signal hypothesis, which proposed that an amino terminal leader sequence targets the polypeptide chain to the microsomes and is then cleaved by a microsomal protease. Many subsequent findings have substantiated this model, including recombinant DNA, experiments demonstrating that addition of a single sequence to a normally unsecreted protein is sufficient to direct the incorporation of the recombinant protein into the rough ER. The mechanism by which secretory proteins are targeted to the ER during the translation, the co-translational pathway, is now well understood. The signal sequence is spanned by 20 amino acids, including a stretch of hydrophobic residues, usually the amino terminus of the polypeptide chain. As they emerge from the ribosome, signal sequences are recognized and bound by a signal recognition particle, SRP, consisting of 6 polypeptides and a small cytoplasmic RNA, 7SL RNA. SRP binds the ribosome as well as the sequence, inhibiting further translation and targeting anti-complex, the SRP ribosome and growing polypeptide chain, to the rough ER by binding to the SRP receptor, ER membrane. Binding to the receptor release SRP from both the ribosomes and signal sequences growing polypeptide chain. The ribosome then binds the protein translocation complex and the ER membrane. A signal sequence is inserted into a membrane chain, both yeast and mammalian cells. The membrane channel, both yeast and mammalian cells. The translocation chain through the ER membrane are complexes of transmembrane proteins called the Sec61 proteins. The process of protein synthesis directly derives the transfer of growing peptide chains through the Sec61 chain and into the ER. As translocation proceeds, the ER sequence is cleaved by signal peptidase and the polypeptides is released into the lumen of the ER. Insertion proteins into the ER membrane. Proteins destined for secretion or residence within lumen of the ER, Golgi apparatus or lysosomes, are translocated across the ER membrane and released into lumen of the ER as already described. However, predestined for incorporation into the plasma membrane are membranes of the ER, Golgi or lysosomes are initially inserted into the ER membrane instead of being related or released into the lumen of the ER. From the ER membrane, they proceed from their final destination along the same pathway as the secretory proteins, ER to Golgi to plasma membrane to lysosomes. These proteins are transported along pathways as membrane components, however, rather than the soluble proteins, as the soluble proteins. Integral membrane proteins are embedded in the membrane by hydrophobic regions that span the phospholipid bilayer. The membrane spanning portions of these proteins are usually alpha helical regions consisting of 20 to 25 hydrophobic amino acids. The formation of an alpha helix maximizes hydrogen bonding between the peptide bonds, and the hydrophobic amino acid side chains interact with the fatty acid tails of the phospholipids. However, different integral membranes proteins differ in how they are inserted. For example, whereas some integral membrane proteins span the membrane only once, others are multiple membrane spanning regions. In addition, some proteins are oriented in this membrane with the amino terminus on the cytosolic side. Others have their carboxy terminus exposed to the cytosol. These orientations of proteins inserted into the ER, Golgi, lysosomal, and plasma membranes are established as the growing polypeptide chains are translocated into the ER. The lumen of the ER is topologically equivalent to the exterior of the cell, so the domains of plasma membrane proteins that are exposed on the cell surface correspond to the regions of polypeptide chains that are translocated into the ER. The most straightforward mode of insertion into the ER membrane results in the synthesis of transmembrane proteins oriented with the carboxy termini exposed to the cytosol. These proteins have a normal amino acid terminal signal sequence, which is cleaved by signal peptidase during translocation of the polypeptide chain across the ER membrane through the Sec61 channel. They are then anchored in the membrane by a second membrane spanning a helix in the middle of the protein. This transmembrane sequence, called stop transfer sequence, signals closure of the Sec61 channel. Further translocation of the polypeptide chain across the ER membrane is thus blocked, so the carboxy terminal portion of the growing polypeptide chain is synthesized in the cytosol. The transmembrane domain then exits the translocation channel laterally to enter the lipid bilayer. The insertion of these proteins in the membrane thus involves the sequential signal sequence that initiates translocation across the membrane and a transmembrane stop transfer sequence that anchors the protein in the membrane. Proteins can also be anchored in the ER membrane by internal signal sequences that are not cleaved by signal peptidase. Proteins that span the membrane multiple times are thought to be inserted as a result of an alternating series of internal signal sequences and transmembrane stop transfer sequences. Protein folding and processing in the ER. For proteins that enter the secretory pathway, many of these events occur either during translocation across the ER membrane or within the ER lumen. One such processing event is the proteolytic cleavage of the signal peptide. As the polypeptide chain is translocated across the ER membrane, the ER is also the site of protein folding, assembly of multi-subunit proteins, disulfide bond formation, the initial stages of glycosylation, and the addition of glycolipid anchors to some plasma membrane proteins. Indeed, the primary role of luminal ER proteins is to catalyze the folding and assembly of newly translocated peptides. As already discussed, proteins are translocated across the ER membrane as unfolded polypeptide chains, while their translation is still in progress. These polypeptides, therefore, fold into three-dimensional conformation within the ER, assisted by the molecular chaperones that facilitate the folding of polypeptide chains. For example, one of the major proteins within the ER lumen is a member of the HSP77, that is, heat shock protein 70, family of chaperones called BIP. BIP is thought to bind the unfolded polypeptide chain as it crosses the membrane and then mediates protein folding and the assembly of multi-subunit proteins within the ER. Correctly assembled proteins are released from the BIP and are available for transport to the Golgi apparatus. Right? And abnormally folded or improperly assembled proteins, however, remain bound to BIP and are consequently retained within the ER or degraded rather than being transported farther along the secretory pathway. The formation of disulfide bonds between the side chains of cysteine residues is an important aspect of protein folding and assembly within the ER. Disulfide bond formation is facilitated by the enzyme protein disulfide isomerase, which is located in the ER lumen. Proteins are also glycosylated on specific asparagine residues, N-linked glycosylation within the ER, while they are translation is still in process. Oligosaccharide units consisting of 14 sugar residues are added to acceptor asparagine residues of growing polypeptide chains as they are translocated to the ER. The oligosaccharide is synthesized on a lipid dolicol carrier anchored in the ER membrane. It is then transferred as a unit to acceptor asparagine residues in the consensus sequence ASNX, SER, serine and 309 by a membrane-bound enzyme called oligosaccharyl transferase. The smooth ER and lipid synthesis. In addition to its activities in the process of secreted and membrane proteins, the ER is the major site of which membrane lipids are synthesized in eukaryotic cells. Because they are extremely hydrophobic, lipids are synthesized in association with already existing cellular membranes rather than in the aqueous environment of the cytosol. Although some lipids are synthesized in association with other membranes, most are synthesized in the ER. They are then transported from the ER to their ultimate destination either in vesicles or by carrier proteins. Smooth ER is abundant in cell types. And that are particularly active in lipid metabolism. For example, steroid hormones are synthesized from cholesterol in the ER. So are amounts of smooth ER are bound in steroid-producing cells, such as those in the testes and ovary. In addition, smooth ER is abundant in the liver, where it contains enzymes that metabolize various lipid-soluble compounds. These detoxify enzymes inactivate a number of potentially harmful drugs, for example, phenobarbital, by converting them to water-soluble compounds that can be eliminated from the body in the urine. The smooth ER is thus involved in multiple aspects of the metabolism of lipids and lipid-soluble compounds. Exported proteins and lipids from the ER. Both proteins and lipids travel along the secretory pathway in transport vesicles, which bud from the membrane of one organelle and then fuse with the membrane of another. These molecules are exported from the ER in vesicles that bud from the ER and carry their cargo first to the ER Golgi intermediate compartment, then to the Golgi apparatus. Subsequent steps in the secretory pathway transport between different compartments of the Golgi and from the Golgi to the lysosomes or the plasma membrane. In each case, proteins within the lumen of one organelle are packaged into the budding transport vesicle and then released into the lumen of the recipient organelle following vesicle fusion. Membrane proteins and lipids are transported similarly, and it is noteworthy that their topological orientation is maintained as they travel from one membrane enclosed organelle to another. For example, the domains of a protein exposed to the cytosolic side of the ER membrane will also be exposed on the cytosolic side of the Golgi and plasma membranes, whereas protein domain is exposed on the luminal side of the ER membrane will be exposed on the luminal side of the Golgi and on the exterior of the cell. While most of the proteins travel from the ER to the Golgi, some proteins must be retained within the ER rather than proceeding along the secretory pathway. In particular, proteins that function within the ER, including BIP signal peptidase, protein disulfide isomerase, and other enzymes discussed earlier, must be retained within the organelle. The distinction between proteins exported from and those retained in the ER appears to be governed by two distinct types of targeting sequences that specifically mark proteins as either one, destined for transport to the Golgi, or two, destined for retention in the ER. Many proteins are retained in the ER lumen as a result of the presence of targeting sequence: lysine, ASP, glutamate, leucine, KDEL in the single letter code. At the carboxyl terminus, the retention of some transmembrane proteins in the ER is similarly dictated by the short C-terminal sequences that contain two lysine residues, KKX sequences. The Golgi apparatus. The Golgi apparatus or Golgi complex functions as a factory in which proteins received from the ER are further processed and sorted for transport to the eventual destinations: lysosomes, the plasma membrane, or secretion. In addition, as noted earlier, glycolipids and sphingomyelin are synthesized within the Golgi. In plant cells, the Golgi apparatus further serves as a site of which the complex polysaccharides of the cell wall are synthesized. The Golgi apparatus is thus involved in processing the broad range of cellular constituents that travel along the secretory pathway. Organization of the Golgi. Morphologically, the Golgi is composed of flattened membrane, enclosed sacs, cisternae, and associated vesicles. A striking feature of the Golgi apparatus is its distinct polarity in both structures and function. Proteins from the ER enter at its cis phase, entry phase, which is convex and usually oriented towards the nucleus. They are then transported through the Golgi and exit from the concave trans phase, exit phase. As they pass through the Golgi, proteins are modified and sorted for transport to the eventual destinations within the cell. Distinct processing and sorting events appear to take place in an ordered sequence within the different regions of the Golgi apparatus. And so the Golgi is usually considered to consist of multiple discrete compartments. Although the number of such compartments has not been established, the Golgi is most commonly viewed as consisting of four functionally distinct regions: the cis-Golgi network, the Golgi stack, which is derived into the medial and trans subcompartments, and the trans-Golgi network. Proteins from the ER are transported into the ER-Golgi intermediate compartment and then enter the Golgi apparatus of the cis-Golgi network. They then progress to the medial and trans compartments of the Golgi stack, within which most metabolic activities of the Golgi apparatus takes place. The modified proteins, lipids, and polysaccharides then move to the trans-Golgi network, which acts as a sorting and distribution center, directing molecular traffic to lysosomes, the plasma membrane, or the cell exterior. Protein glycosylation within the Golgi. Protein processing within the Golgi involves the modification and synthesis of the carbohydrate proteins and carbohydrate portions of glycoproteins. One of the major aspects of this processing is the modification of the N-linked oligosaccharides that were added to proteins in the ER. As discussed earlier, in this chapter, proteins are modified within the ER by the addition of an oligosaccharide consisting of 14 sugar residues. 3 glucose residues and 1 mannose are then removed while the polypeptides are still in the ER. Following transport to the Golgi apparatus, the N-linked oligosaccharides of these glycoproteins are subject to extensive further modifications. Proteins can also be modified by the addition of carbohydrates to the side chains of acceptor serine and threonine residues with specific sequences of amino acids, O-linked glycosylation. These modifications take place in the Golgi apparatus by the sequential addition of single sugar residues. The serine or threonine is usually linked directly by an acetyl-galactosamine, to which the other sugars can then be added. Proteins destined for incorporation into lysosomes are linked by the mannose 6-phosphate. Lipid and polysaccharide metabolism in the Golgi. In addition to its activities in processing and sorting glycoproteins, the Golgi apparatus functions in lipid metabolism, in particular in the synthesis of glycolipids and sphingomyelin. As discussed earlier, glycerol, phospholipids, cholesterol, and ceramide are synthesized in the ER. Sphingomyelin and glycolipids are then synthesized from ceramide in the Golgi apparatus. Sphingomyelin, the only non-glycerol phospholipid in cell membranes, is synthesized by the transfer of a phosphorylcholine group from phosphatidylcholine to ceramide. Alternatively, the addition of carbohydrates to ceramide can yield a variety of different glycolipids. In plant cells, the Golgi apparatus has the additional task of serving as a site where complex polysaccharides of the cell wall are synthesized. The plant cell wall is composed of 3 major types of polysaccharides. Cellulose, the predominant constituent, is a simple linear polymer of glucose residues. It is synthesized at the cell surface by enzymes in the plasma membrane. The other cell wall polysaccharides, hemicellulose and pectins, however, are complex branched chain molecules that are synthesized in the Golgi apparatus and then transported in vesicles to the cell surface. The synthesis of these cell wall polysaccharides is a major cellular function, and as much as 80% of the metabolic activity of the Golgi apparatus and plant cells may be devoted to polysaccharide synthesis. Protein sorting and export from the Golgi apparatus. Proteins as well as lipids and polysaccharides are transported from the Golgi apparatus to their final destinations through the secretory pathway. This involves the sorting of proteins into different kinds of transport vesicles, which bud from the trans-Golgi network and deliver the contents to the appropriate cellular locations. Some proteins are carried from the Golgi to the plasma membrane by a constituted secretory pathway, which accounts for the incorporation of new proteins and lipids into the plasma membrane, and as well as for the continuous secretion of proteins from the cell. Other proteins are transported to the cell surface by a distinct pathway of regulated secretion or are specifically targeted to other intracellular destinations such as lysosomes in animal cells or vacuoles in yeast. Proteins that function within the Golgi apparatus must be retained within that organelle rather than being transported along the secretory pathway. In contrast to the ER, all the proteins retained within the Golgi complex are associated with the Golgi membrane rather than being soluble proteins within the lumen. The signals responsible for retention of some proteins within the Golgi have been localized to their transmembrane domains which retain proteins within the Golgi apparatus by preventing them from being packaged in the transport vesicles that leave the trans-Golgi network. In addition, like the KKXX sequences of resident ER membrane proteins. Signals in the cytoplasmic tails of some Golgi proteins mediate the retrieval of these proteins from subsequent compartments along the secretory pathway. They constitute a secretory pathway which operates in all cells, leads to continual unregulated protein secretion. However, some cells also possess a distinct regulated secretory pathway in which specific proteins are secreted in response to environmental signals. Examples of regulated secretion include the release of hormones from endocrine cells, the release of neurotransmitters from neurons, and the release of digestive systems enzymes. From the pancreatic acinar cells, proteins are sorted into the regulated secretory pathway and the trans-Golgi network, where they are packaged into specialized secretory vesicles. These secretory vesicles, which are larger than other transport vesicles, store their contents until specific signals direct their fusion with the plasma membrane. For example, the digestive enzymes produced by pancreatic acinar cells are stored in secretory vesicles until the presence of food in the stomach and small intestine triggers their secretion. The sorting of proteins into the regulated secretory pathway appears to involve the recognition of signal patches shared by multiple proteins that enter this pathway. These proteins selectively aggregate in the trans-Golgi network and are then released by budding as secretory vesicles. The best characterized pathway of protein sorting in the Golgi is the selective transport of proteins to lysosomes. As already discussed, luminal lysosomal proteins are marked by mannose-6-phosphates that are formed by modifications of the end-linked oligosaccharides shortly after entry into the Golgi apparatus. A specific receptor in the membrane of the trans-Golgi network then recognizes these mannose-6-phosphate residues. The resulting complexes of receptor plus lysosomal enzyme are packaged into transport vesicles. Destined by lysosomes, lysosomal membrane proteins are targeted by sequences in their cytoplasmic tails rather than by mannose-6-phosphate. Lysosomes. Lysosomes are membrane-enclosed organelles that contain an array of enzymes capable of breaking down all types of biological polymers, proteins, nucleic acids, carbohydrates, and lipids. Lysosomes function as a digestive system of the cell, serving both to degrade material taken up from outside the cell and to digest obsolete components of the cell itself. In their simplest form, lysosomes are visualized as dense spherical vacuole, but they can display considerable variation in size and shape as a result of differences in the materials that they have taken up for digestion. Lysosomes thus represent morphologically diverse organelles defined by the common function of degrading intracellular material. Lysosomes contain about 50 different degradative enzymes that can hydrolyze proteins, DNA, RNA, polysaccharides, and lipids. Mutations in the genes that encode these enzymes are responsible for more than 30 different human genetic diseases, which are called lysosomal storage diseases because undegraded material accumulates within the lysosomes of affected individuals. Most of these diseases result from deficiencies in single lysosome enzymes. For example, Gaucher's disease, the most common of these disorders, results from a mutation in the gene that encodes a lysosomal enzyme required for the breakdown of glycolipids. An intriguing exception is eye cell disease, which is caused by a deficiency in the enzyme that catalyzes the first step in the tagging of lysosomal enzymes with mannose-6-phosphate of the Golgi apparatus. The result is a general failure of lysosome enzymes to be incorporated into lysosomes. All of the lysosomal enzymes are acid hydrolases, which are active at the acidic pH about 5 that is maintained within lysosomes, but not at the neutral pH about 7.2 characteristic of the rest of the cytoplasm. The requirement of these lysosomal hydrolases for acidic pH provides double protection against uncontrolled digestion of the contents of the cytosol. Even if the lysosomal membrane were to break down, the released acid hydrolases would be inactive at the neutral pH of the cytosol. To maintain the acidic internal pH, lysosomes must actively concentrate hydrogen ions, protons. This is accomplished by a proton pump in the lysosomal membrane which actively transports protons into the lysosome from the cytosol. This pumping requires expenditure of energy in the form of ATP hydrolysis, since it maintains approximately 100-fold higher proton concentration inside the lysosome. Endocytosis and lysosome formation. One of the major functions of lysosomes is the digestion of material taken up from outside the cell by endocytosis. However, the role of lysozymes in the digestion of material taken up by endocytosis relates not only to the function of lysosomes but also to the formation. In particular, lysosomes are formed by the fusion of transport vesicles and buried from the trans-Golgi network with endosomes, which can contain molecules taken up by endocytosis at the plasma membrane. Right, so that sums up a certain portion of the activities of mitochondria. The remaining portions will be in the next lecture following very shortly. So that's it for now. Thank you.